Hey guys, welcome to the second lesson on mitosis and cell division. I have three goals for you guys for this video. The first thing is I want you guys to be able to explain the steps of mitosis. Secondly, I want you guys to be able to label what happens during each step. And finally, I want you guys to be able to describe what's actually happening to the DNA during mitosis. Now, this may seem really easy, but boy, there's a lot of memorization to go on here. So I would highly recommend that you put the attention needed into the activities I've created for you here because they will help you to understand what is going on. So, mitosis. You hopefully remember this term from the last lesson. Mitosis is just a really fancy way of saying cell division. And all I'm saying here, it's taking one cell, so I have a little happy cell here, and it's making, that's a terrible arrow, two new cells. That's all mitosis is. It's taking one cell and making two new cells. They're basically just exact copies of that guy right there. So I better give him the smiley face too, because he's a very happy cell. Don't know why. So there are four steps to mitosis. There's prophase, metaphase, anaphase, and telophase. Now I always tend to remember these as PMAT. I don't know why it works like that for me, but it works. If you sit there and try to turn it into a little bit of an acronym, it makes it a lot easier to remember this stuff. So before we go into the steps of mitosis, let's first talk about what's going on with the DNA a little bit. So, if you remember from last lesson, chromosomes are these long strands of DNA that you will find inside of cells that hold the DNA, and these are DNA and proteins. However, during the S phase of the cell cycle, the DNA replicates, and you get one. Rather than having one chromosome, you actually begin to have two chromosomes that are exact copies of each other. So these two chromosomes are exactly the same. They are identical in every single respect. And we call them sister chromatids. If you want to know what the word chromatid means, chromatid is just a fancy way of saying one chromosome. So they're sister chromatids. They are exact copies of one another. And that's what happens. You have these two pieces of DNA, and they're an exact copy of the original one right here. Now, here in the middle, you can't see it on this diagram, but there's going to be a central thing here called a centromere. A centromere is basically like glue. It's going to glue these two chromosomes together so they don't leave each other. Because you don't want them flying apart, you want them to be together until you need to separate them later. So, here is the diagram that, I'm sorry guys, will be a challenge. You are going to have to put your effort into this if you're going to want to learn this for the activities and the assessment. So, the very first step in the DNA cycle is called interphase. Now, that actually is not part of mitosis. This is just part of the cell cycle. But during this, the DNA replicates. Now, remember, this happens during the S phase of interphase, or the cell cycle. Now, the very first step in the cell cycle is called prophase. This is when a couple things happen. First off, the nuclear membrane disintegrates. That's this fancy looking blue purple thing right here. And this thing falls apart. As you can see right here, it disappears. The second thing is that the nucleolus disappears. That's just a fancy term for this little object right here. It also disappears. That nucleolus holds the DNA, so you're going to want to see, and also involved in protein production. But you don't have to know that. The chromosomes condense. What that means is the chromosomes come together just like you see down here. They turn into these little like bow tie looking things, or maybe you can call them as X-wings from the Star Wars series. The chromosomes come together, and what's called the mitotic spindle begins to form between the two poles. Now you can't actually see it here, but basically what that is what you're looking at down here. These right here are the mitotic spindles. They're basically like Spider-Man's webbing, where they're going to grab onto the chromosomes and move them around. They kind of act like the movement thing to keep the chromosomes to where they get the chromosomes to move where they want them. Now, the second phase of mitosis is called metaphase. Really, all that happens during metaphase is that the chromosomes are going to attach to these little strings here called mitotic spindles, which you can see right here in this little box, and they align in the center. So these mitotic spindles are going to try to get these chromosomes to line up here in the middle so they can be easily divided between the two sides. And a cool part here that you know is that oftentimes, if there's a failure in this process and these things can't get to the middle, the cell will actually die and self-destruct. Or, even more terrifying, the cell can become cancer, which we'll talk more about in the next lesson. Now, after metaphase, as you can see here, all the chromosomes have lined up here in the middle. What happens now is that the microtubules begin to shorten. You're probably thinking to yourself, what does that mean? Oh my lord, oh my. Well, these things right here, the mitotic spindles, are made out of microtubules. And if you remember that term from last year, the microtubules are just little structures inside the cell that give it its shape. So these chromosomes are attached to microtubules and they start to get pulled apart. So during anaphase, what you see is this right here. You see the chromosomes getting split in half. Now you might have noticed that one chromosome went over here and one chromosome went over here. These two things are sister chromatids. They're being split apart and they're being moved to different sides of the cell. Now, the very last phase is called telophase. This is basically returning everything back to normal. So as you can see, the nuclear envelope is returning. So this little purple part here called the nuclear envelope is coming back. The nucleosome, or so the nucleolus is coming back. The sister chromatids are finding their homes in their new nucleus. And if you notice right here, there is a pinch happening in the cell. The cell is being pinched by the mitotic spindle. And that pinch right there is what we call cytokinesis. If you look at this picture right here, what you see is the cells pinching off and becoming two new brand cells. And that's what cytokinesis is. Cytokinesis is just the splitting off of the two cells, and it's beginning to form during telophase. Now, this is going to take some effort to learn, guys. It took me a lot of effort to figure it out for myself, and I still have to give myself refresher courses on this every now and then. So do your best, and just remember to look at it as a series of steps. Just remember that the DNA has to be split in half so both cells get an equal share. So as long as you know the cell has to get an equal amount of DNA on both sides, you can kind of figure out logically what's going on from step to step to step. Now, here's some tips to understanding what's going on in the phases and how you're going to begin to look at them for the assessments. First off, really look to see where the chromosomes are, because if you look back at here, what the chromosomes are doing is really important to understanding the various phases of mitosis. So if you want to know what's happening, look at the chromosomes. Second, look at the centriole is formed. The centriole is this weird looking thing right here because the centriole, if you look at prophase, it's not there yet. If you look at metaphase and anaphase, boy, it's there all right. But if you look at telophase, it's gone. So the centriole is present in two of the phases, but not in the other two. Finally, look to see what the sister chromatids are together or not. If you look here, here, and here, the sister chromatids are all together. However, in anaphase and telophase, the sister chromatids are being split apart. So that's kind of a way to remember what's going on. So remember what's going on at each step. Now, a couple other just miscellaneous things. I've already talked about them, but this PowerPoint will give you a way to see it. The spindle apparatus, I talked beforehand, is going to be this weird little purple stuff you see right here. And its basic job is just to pull the fibers they pull the sister chromatids apart. They pull those two chromatids apart, so rather than having one chromatid that looks kind of like this, the spindle apparatus is going to make it two separate chromatids like this. Now, cytokinesis, that's just a fancy way of saying when the cell splits in half, and that is this part right here. And that's it, guys. That's all the stages of mitosis. Like I said, this is challenging. This will be difficult, but I know you guys can do it. So, just make sure there are 4 stages to mitosis. I try to remember them as PMAT. It sounds weird, but it works. I have one more acronym I placed in one of the other things I gave you, and that should give you an idea. I think it was like people meet apples today. I forgot what it was, but there are some cool acronyms out there for remembering the phases of mitosis. It's a spindle apparatus that pulls the sister chromatids apart, and each step in mitosis is based upon what is happening to the chromosome. So if you try to remember what's going on, just remember the chromosomes are what is important here. And that's it, guys. Thank you for watching the video, and I hope you guys do well with the activities.